But how do we measure whether or not they're successful? To give us some pointers, Professor Laurent Servais, who's Professor of Pediatric Neuromuscular Diseases at the Oxford University and Professor of the University of Liège.
Thank you very much. I should, like Francesco, thank the organizers. But I will not. Because, guys, asking me to speak about Volker and Francesco, asking me to speak about the German robustness spiced by artificial intelligence, asking me to speak about the consensual wisdom surrounded by Mediterranean accent of Francesco. How can I do that? And what does it remain to me except being provocative, which I hate, to be honest. So, I will be Belgian. And the second reason why I do not thank you is about the title. How do we measure success in a clinical trial? That's me preparing my talk and thinking about the title. So I use an outcome measure that I will call the motivation meter. So there are different levels. It starts with, I will say that I have COVID and that I can't do it. And it goes up to, that's going to be the best talk ever of the history. And I was stuck with a middle level, I don't like the title. So, how do we measure success in a clinical, but who has thought about such a title? Because it's a question to start with, and this question brings another question, which is what do you mean by success? What is a success, right? And so let's imagine a drug, okay? Let's call it la Muntonin, la buona medicina di Papa Francesco. And so the question is, what will make a clinical trial successful with the Muntonin, right? Is it successful because it shows that it works, even if it does not work? Or is it successful because it answers clearly the question? And for me, and I'm sorry if I missed the title of the talk, but success in a clinical trial means to answer the question that we ask, and not to show by any possible way that it works, even by dividing by the age of Francesco, multiplicating by the postcode of Newcastle, and then comparing Francesco and Volker, and then eventually to show that it works because you compare with people who were walking when I was a baby myologist. So for me, we need to answer a question. We do a trial because we have a question about safety, about efficacy, and what makes success is when we answer the question. And I'll continue being slightly provocative because I think that's why we are here, is to discuss and to sometimes bring new ideas, new concepts. We've heard a lot about different phase 3 trials during the first talks. Have we heard something about being sure of the results? I've heard, well, the trial has missed the primary endpoint, but if we look at the secondary endpoints, it works. Well, the trial has missed the primary endpoint, but if we look at the patients who were not included during the COVID, but the patients who are propensity score, and compared with, but at the end of the day, did we really answer the question? So we end up with trials with more questions and more doubt than before the trials. So for me, the definition of success will be to answer the questions clearly, and we need to face the answer. And we need to accept that the answer is, maybe a drug does not work, and that's it. And then you say, come on, be slightly more smart. It's not because you do not reach the primary endpoint that the drug does not work, and it's true. But if we need 500 patients per group during 10 years, does it mean that we need to expose our kids, our boys, our patients? And does it mean that we need to pay for a drug if we need so many patients and so complex outcome measures? So that's for me open questions. So how do we measure success in a clinical trial? I will take this definition. A clinical trial is successful if it clearly and honestly answers the questions that we were trying to answer. That's my definition of success. Okay, this is the classical vision of a clinical development with phase 1, phase 2, phase 3. Phase 1, you try to answer the safety, phase 2, the dose, phase 3, the efficacy. In the rare disease, most of the time we have phase 1, 2. And what we see so often in the field, not only in Duchenne, but in the field is phase 1, 2, sounds safe. Look, it works. There is a surrogate biomarker and historical control. When you match, it works. Let's go for early approval, and let's increase the stock exchange rate. So, and then we go to a phase 2 trial. Doesn't work, but maybe, there is a imbalance between groups, maybe the outcome measures, maybe it was too short, maybe the population, maybe COVID-19. And that's a long story that we have heard a lot during clinical development in the field, in Duchenne or in spinal muscular atrophy. How can we go out of this? How can we really answer the questions? Okay, let's go back to phase 1, 2 trial. How can we answer the question, is this drug, lamuntonin, is it safe? And what is the dose? The question that we ask is the target engagement, and Francesco made a point by showing the different amount of dystrophin we express. And then, look, the number of comma, signal of efficacy, maybe. It's a signal. A phase 1, 2 is not done to demonstrate efficacy. Keep this in mind. It's impossible to design a trial that answers all the questions together. A phase 1, 2 is done to answer the question of the safety, to a certain extent of the dose, and to demonstrate the target engagement. And that's it. Efficacy will come later. So, some consideration on phase 1 trial objective. Safety. It's not only measured by the occurrence of adverse event or serious adverse event, but also by biological tests. And sometimes we can clearly see that even if the patient has no clinical problem, because of, troponin I that increase, because of creatinine that increase, because of subtle renal abnormalities, we end up by saying it's going to be difficult to bring it to a large population. And again, be careful about any statement of efficacy. And this is really something I would like to reiterate. We should, as a community, be extremely careful and remind that phase 1 is a phase 1. So, phase 1 are not powered and designed to demonstrate efficacy. There is, most of the time, no control group. Or only some patients, 3 patients on placebo for 10 patients on treatment. It's not a control group. And then I would like just to speak about this propensity score analysis. Most of the time, we see a lot of such analysis. I've never seen, I'm lying as usual. I've seen 1 or 2 studies that are negative, but most of them are positive. Each time that we have propensity score analysis, it's positive. And we see, that's amazing, we have something. But have you seen, guys, the statistical analysis plan of such analysis? So it means that before running this analysis, are we aware that we are running this analysis? Or do we see just those that are positive with some cherry picking? Do we know a priori which is going to be the control group? Do we know a priori which is going to be the main outcome, the primary outcome, the secondary outcome? Do we know a priori, are we going to match the patients? I've never seen any of these questions before we got the results. And that's a problem. That's really a problem of transparency. Because I'm pretty sure that you can take any set of data and find a way to match it and find a way to show that it works. If we do not show before the analysis, the statistical analysis plan, the population we're going to match with, the outcome measures that we will choose, we just cannot believe the results. We need to see all of this before the analysis is done. Otherwise, it can be just cherry picking. We have dozens of examples of very promising drug in phase 1 with positive results with propensity score analysis that failed in phase 3. And we need to be careful of that. And especially when we speak with families, when we speak with patients, we need to raise realistic expectation. You cannot imagine how many emails I receive as a physician from everywhere in the world after any press release, after any public presentations of results, telling me, "Doctor, I've heard about it. Is it possible to get this drug in your clinic?" And raising unrealistic expectations in the community, in my opinion, is extremely dangerous because it leads many families around the world to take the wrong decision for them, for the child, and for themselves. Okay, let's now move to a phase 3 trial. Is la buona medicina di Papa Francesco working or not? A successful trial is a trial that will clearly and honestly answer the question. That was my point. How are we going to measure it? And I'm not going to give you a long explanation about outcome measures, and I'm not going to explain you here in London what is the North Star, right? It's like preaching in a choir. It's like going to Roma and explain what is the Bible. It's impossible, you know. So I'm going to explain you more generally what are the outcomes and what has been our work in this. So the question is, why can we fail in a phase 3 trial? First, because it's not powered enough, because we have not enough patients, right? And it's true that if we have only, 20 patients per group, we have less chance to show a statistical significance than if we have 100 of patients per group. But it comes with a price to pay. If you need 200 patients per group to demonstrate a difference, it means that the difference is small, right? And if this difference is small, is it going to justify the burden and the cost of the treatment? 2, is it the right population? Have we thought about the right population to study the drug? And I would like to bring here a concept of window of opportunity. Are we sure that we're not coming too late? And I think Francesco made an excellent point, as usual, on the age of the patients. And I'm not sure that there are not some drugs that we probably underestimate the efficacy because they have been delivered too late, right? And we're not able to measure anymore that there is a benefit. Look, for instance, for spinal muscular atrophy. Our nusinersen risdiplam gene therapy is incredibly efficient if you give it at birth, but if you give it later, you will struggle with large core to demonstrate efficacy. Then you have the questions of the right outcome. And to be honest, some years ago, it was always, the usual suspect was, we did not pick up the right outcome. But I think that the questions of the drug itself, the population and the power is also key. And then you may have the problem of not being properly conducted. Are we sure that there is no unblinding? Are we sure that we can really compare our patients with the controls? Are we sure that there is not any difference in the way we have managed the patients in the controls? When it comes to gene therapy, are we sure that we are giving the same dose of steroids to patients and controls? And many other factors. So again, in this concept of thinking the success as our ability to clearly demonstrate the efficacy, let's think a priori to all the reasons why we could have a failure. And a failure is not to show that a drug does not work. A failure is not being able, after a phase 3, to demonstrate that the drug works or does not work. So, how do we measure efficacy? We have strength. And it's quite obvious that strength is something that we want to increase in muscle conditions. But the question is, how strength is related to function? So Francesco made the point that he wanted to run with Volker. And to be honest, I will buy the ticket to see the race. Let's imagine that I give 10 newtons more to Francesco because, as he rightly said, he needs a kind of a compensation. Is this 10 newtons, 100 newtons in terms of strength that I will give to Francesco because he deserves it? Is it going to translate into any functional benefit, and this is not clear. And the relationship between the strength and the function is real, but absolutely not linear. It means that when you're very weak, a little bit of strength can increase your function. If you are very strong, a little bit of strength will not change your function. We can measure the performance. The performance is measured by, for instance, all the time test, and we know now the infamous 6-minute walking test that we've practiced, all of us, which, to be honest, is probably not as bad as we used to think, in my personal opinion. But the problem is the motivation, and you know the story of this group of kids for which we asked to make a 6-minute walking test, so to walk as much as they can, and then they were randomized. 50% of them were asked to do it again and to do better, and 50% of them were asked to do it again and to do better, and if they were successful, were promised $50. The group that was promised $50 actually defeated the other group by 80 meters, which means that with $50 you can gain 80 meters, so nearly 3 times the MCID on the 6-minute walking test. So don't pay $300,000 per year, guys. I mean, with $50 you can do the job. Just to say, we need to pay attention to this test because of the aspect of motivation and the fact that this is a point assessment. It means that if the child is tired, if the child is bored, if the child does not want to cooperate, if this child comes from very far away because he lives in a remote place of the country, he comes to the capital, there are plenty of things to see, the Eye of London, Marie Tussauds, and then the aquarium, and then the day after he's exhausted and he runs the test. So we need to pay attention to that. And then we have the very short tests, like the Forster climbing tests, which are a little bit coming back now under the spot, but let's keep in mind that there is a very narrow range during which there is a linear evolution of this test. And when we talk about 1 second or 2 seconds, what is the component of strength and what is the component of reflex time of the physiotherapist and the patient themselves? And then we have the North Star. And again, I'm not going to explain you guys what is a North Star here in London. I feel absolutely incompetent for that. This scale scores on 34. There were different approach to refine it, but I like to take the linearized North Star to consider shift analysis. But today it's mostly the main scale that is used. And I think that the beauty of this scale is certainly the amount of data that has been collected and the systematic aspect across a large network in the UK. But now I'm going to speak a little bit about the work of my team. So the challenge that we had was mostly identified when I was in Paris, what I called Eiffel Tower syndrome, in which our patients were exhausted when they come for the test because they had been visited the Eiffel Tower. So the good news is that when they come back 6 months later, they're still exhausted because they went to Euro Disney. So we bring the question that probably the problem is that we're just looking at point assessment, and that if rather than to look at the point assessment at a very specific time period, we could look at the area under the curve, we could be much more powerful in defining a difference between 2 populations. So it came also with a very funny experience that was done during a protocol research in which that was driven by Francesco, led by Francesco, in which Volker and I were shy co-investigators, Skip and MD, and during this research we placed an accelerometer on patients, and we saw that when they leave the hospital and they are happy not to see me again, they walk faster than during the 6-minute walking test, which is a problem. It brings some questions about the 6-minute walking test. And of course, the reflex time of the Forster. So, of course, you could think, okay, that's the place of wearable, and that's what we did during our job with in the Skip and MD, and we did things that are ugly. You know, a first prototype that was ugly, a second one that was terrible, and otherwise that was not beautiful, and then it was still ugly, and then progressively it becomes better and better, and we eventually identified SV95C. So that's someone in the subway in Paris. She has a mask, not because of the COVID, but because the odor of urine, the smell of urine in the subway of Paris. I'm not French. And we can see clearly the stairs and every single step. So the concept is to measure every single step of the patients and to identify them, and then to take the 5% most rapid strides. It has been qualified by EMA as a primary outcome measures. And then you could say, okay, but it's so variable, Guy. I mean, when you were in Paris, you used to say when the French are on strike, they walk more. And now that you came in the UK, you say when it pours, British people walk less. When it simply rains, they walk more, which is true. But the point is, do we walk a different way? And if we walk with we can maybe walk less, but the way we walk is not so different. So another point with all these home-based measures is that, yes, you may have a huge variability on a day-to-day basis. But if you average on 3 days, then on 9 days, then on 10 days, you decrease the variability, right? And it allows to detect, to see early decline, and it has been demonstrated on the placebo group of Roche to avoid the risk of false negative. I think that Volker has shown this data already. And also in the exon skipping drug to show an early benefit of treatment. But more importantly, and that's how I would like to finish this discussion, maybe to assess a population that we probably should assess much more, and this is a plea for the community, is the toddlers. May I ask a question? Who has a toddler at home? You have a toddler. What's the name of your toddler? Kate? Kate. So how old is Kate? Kate. 3.5. If you ask Kate to do something exactly the way you want him to do something, is he going to do it? Absolutely not. If you ask him to do the same thing because you want him to be reliable, and you want him to do the things the way that Marion Main wants him to do, or Michel Egan wants him to do, is he going to do it? No. That's the point. So the toddler are not really reliable. But if you just put a sensor on the toddler and tell the toddler, you know what, I don't care. I don't care. Do whatever you want. Live your life. Leave me alone. And I'm going to record you. Big Brother is watching you, my dear little toddler. And I will just follow your performance. The beauty is that if you do so, first, the toddlers, they love this type of device. You just tell them that it's a magic watch and that's it. It's highly reliable, 0.99, which is insane in terms of reliability. And the beauty is that sooner or later, the toddlers show their top performance, and we can clearly see the difference at the age of 18 months between the toddlers with Duchenne and the toddlers with control, and we can clearly see what happens when we give steroids. It means that if you don't ask a toddler to do anything special, he is highly reliable, and sooner or later he makes his top performance. So now my PhD student tells me, stop asking me to do anything, I will be highly reliable and I will make my top performance. But it works only for toddlers, not for PhD students. And I think that we really need to think about this population, because we need to think also in terms of window of opportunity. Sometimes we are unable to show the difference in all the population, but let's think, are we not missing the window of opportunity to leverage the efficacy? And with that, I would like to conclude with some general context. One measure is not enough. We need a primary, we need secondary, we need tertiary outcome measures. And it's extremely important. First, because all these measures do not assess exactly the same. I talk about the function, about the performance, the strength, or the activity. We need all these different portfolio of measures, and we need to hierarchize, because we need to understand what is the primary, the secondary, and the tertiary. And that's extremely important, because normally we're supposed to look first at the primary, and then the secondary, and then the tertiary outcome, and to look at this way and not another one. Not too many measures, because too many measures kill the measures. You get your patients exhausted, and we have some trials with so many measures that I'm pretty sure that the last measure we do in these kids are not reliable. So the other thing is that if you have 20 measures and only one that is statistically significant, you can say it's by chance, right? So let's try to have enough measure, but to tailor the measures to those who have a decent chance to be positive. It needs to be appropriate, and it needs to measure what we want to measure. And finally, we need to work on these outcome measures with the family and with the patients, because we need the collaboration not only to construct, to build these outcome measures, but also to understand the need for reliable assessment. And with that, I would like to thank my team in Oxford, my team in Liège, and finally the organizers, because thank you so much for inviting me.